Amelie, Gubitz, you'll take things off.
Okay, good afternoon, everybody. So our group was tasked to develop recommendations for the transition between preclinical studies and clinical trials. And of course, the key question is, what type and what level of preclinical data do we need to feel confident that we should start a clinical trial? So, of course, all of this is governed by the regulatory process, and certainly the regulatory agencies will give us the green or red light whether a therapeutic agent can go into clinical testing. But what our group really wanted to do is to focus on scientific premise. And I think this is a very important and exciting area. We are dealing with a rapidly evolving landscape. I think over the past few years, many new genetic ALS animal models have been developed, and we are also now using patient-derived cell lines. And I think sort of the development of iPSC technology is really a paradigm shift across biomedical research, and certainly ALS researchers are harnessing that technology. So our group was assigned 5 questions, and actually each of the topic leads are going to give a brief presentation. We had very good discussions around our topics. I think we didn't always fully agree. Our sort of views are a little bit on a spectrum, but I do think that we developed many recommendations with a lot of substance. And I should mention that there's a little bit of overlap between the questions and our recommendations. So I think one task for our group after the workshop is to consolidate some of the recommendations that we made for each of the research questions. And finally, I would like to thank all my team members. They got many nagging emails from me, and they all stepped up to the plate and got the job done beautifully and masterfully. And I'll hand over to our first speaker, and that's Brian Traynor.
Thank you, Amelie. Pardon me, I know I've got a lozenge in my mouth. My throat is going a little bit this morning. So I'll go up to the first slide, please. Okay, so the first question is, okay, thank you. First question is, do we need models with efficacy signals, or can we identify drugs solely based on disease mechanisms and biological hypothesis? And I'm presenting this on behalf of our group, which includes Steve Barron, Stan Appel, Jen Sobu, and myself. I would say that as Amelie has pointed out, there is some overlap between the points that we're making and what you're going to hear in the subsequent talks, which is great. I think that there was also quite substantive debate among ourselves as to how we were going to present this. So we're very open to input, and we are looking forward to questions and some points being raised. Okay, so what's the background to this? Well, first and foremost, the demonstration of efficacy in disease models is and I think everybody will agree on this point, is a traditional component of drug discovery. Now, that's partly because it's a prerequisite from the regulatory requirements, FDA and so forth, and European regulatory, but also because the preclinical efficacy data helps us to prioritize which drugs we bring forward for clinical trials. Because remember, there are a paucity of patients, and therefore a limited number of trials that we can do at any one time. Nevertheless, the development of drugs for ALS has been hampered by animal models that fail to reflect the complete spectrum of phenotypes represented in this polygenetic disorder. Now, I struggled over the wording of that, and that was the most diplomatic way I could actually phrase that. So anybody who wants to correct me, that's absolutely fine. This compounded with an inconsistency in the design and execution of preclinical studies involving animals across the field, and has resulted in a sort of a difficulty in translational efforts with high attrition rates and no examples of really truly successful translation. In parallel with, so that's kind of a little bit the bad news. The good news is that in parallel with that, there's been fundamental improvements in our ability to model patients using cell-based models, and I'm specifically referring here to iPS cell lines, together with 3-dimensional tissue models and other complex in vitro systems. Okay, the rest of the background. At the very least, I think that those models, and I think that I speak on behalf of the group, provide a means to confirm that a candidate drug is engaging with its target. Though, again, we've got to recognize that some of those cell models can't effectively model the very complex pathophysiology that we know is involved in ALS. And the key question for the field, and something that we all try to think about and tackle, is whether these in vitro-based systems now represent a valid and sufficient alternative to the use of animal models for preclinical efficacy testing. So that's the background. And in terms of the actual points that we want to raise. So rationale 1. Disease-relevant animal models remain the gold standard method for screening candidate drugs for target engagement, to look for efficacy signals, and to prioritize drugs for human studies. The outcome will vary by model. So what are our recommendations? Well, the first one is, and this is two slides on this recommendation, on this particular rationale. A, the development and validation of disease-related animal models should be prioritized within the field. I think we all agree that we need new animal models, and even this week there are some additional ones coming out. B, outcomes should be selected for each animal model based on their individual natural history of disease, and may involve measurements of survival, motor function, pathology, neurophysiology, function, or other parameters as appropriate. We want it to be as broad in our sweep there as possible. We're not suggesting that any one of those is the key to it. We just want to encompass as many of them, and more make the point that the outcome measure has to be tailored to the animal model itself. C, investigators who develop an animal model are encouraged to provide detailed parameters and guidelines for outcome measurements. And this will allow other researchers to replicate their findings and will improve consistency and reproducibility across the field, which is, of course, something that we've all been concerned about over the last few years. D, efficacy data for animal models should include a dose-response curve whenever feasible. Very important, we think. In the absence of positive efficacy signal, this is where I think we're going to have some debate, a robust dose-response curve based on a pharmacological or pharmacodynamic outcome in the target tissue with a strong biological and therapeutic rationale may be sufficient. F, lead compounds should be validated in multiple models if possible. Okay, rationale number 2. Our ability to model human disease using iPS cell lines and complex in vitro systems is rapidly evolving. Such patient-derived cellular models may offer a valid means of modeling ALS pathogenesis and may provide a means to test drug efficacy by looking for molecular or pathological changes. So what are our recommendations? A, the development and validation of appropriate iPS and other cell models should be prioritized within the field. Fair to say that that's actually already happening. B, the robust phenotypic characterization of such models is imperative. C, molecular and phenotype endpoints for these systems should be validated using multiple sources of the model of interest. So what do we mean by that? Well, for example, iPS cell lines derived from multiple patients with the same genetic mutation. In other words, for example, multiple iPS lines derived from C9 or 72 patients. D, utilization of cell-based systems for lead discovery and lead optimization requires statistically valid assays using multiple sources for the model of interest. So in other words, iPS cell lines derived again from multiple patients with the same genetic mutation, something that we're harping on about again and again. E, lead compounds should be validated in multiple models. F, dose response should be rigorously characterized for lead compounds in these systems. And the last point is, prior to clinical translation, lead compounds need to be characterized using animal models for tolerability, pharmacokinetic and biodistribution, and target engagement. So I'm going to stop there. Perfect on time. Rushed it a little bit because I'm aware that we've got 45 different recommendations that we have to go through, but I'm opening it up for debate at this stage. Thank you.
Brian, that was great. Brian, that was great. It was a good start. Right from the outset, I completely disagree with the concept of gold standard. Right. There's no reason whatsoever to make that a gold standard. Gold standard usually means it's been validated, it works, and there's no data to suggest that. Many of the drugs we use in neurology were not based on animal models. They're based on knowing a target engagement, and they don't require whole animals. I would say we recommend using animal models as the toolbox, but personally I would take gold standard. I do not believe
would current standard be a better word?
I don't know if it's a standard. I would just say it's a tool. It's an important tool.
But it is what it is. In the field, it is what is used at the present time to determine what goes forward for clinical trials. Would you agree?
Looking forward to think about how we change that.
Yeah, no.
May I make a comment here? This is not the time to have a dialogue about a point and make a decision. This is the time for us to give input that will hopefully be very graciously received and integrated into a more final form for recommendation.
So the next one then is, when you talk about IPS lines, you should probably include isogenic.
Sure. Yep. I think that's very reasonable.
Yes, you did not include iPS cell from sporadic ALS. I think that was a great advantage of the iPS cell to better understand the sporadic ALS. So while limiting this recommendation to the familial one, those with the genetic mutation.
I'm sorry, Jean-Pierre, there was a terrible echo.
The sporadic ALS, there's no mention to work also with iPS cell from sporadic ALS. So you only limit yourself to the genetic one. And that's a fantastic tool to investigate what's going on with the sporadic ALS. I think it should be included.
I agree with you. I think that we tried to cover that by talking about complex pathophysiology, but I agree putting in sporadic specifically would be a good idea.
Yeah, so I think in general our group did not exclude sporadic lines whatsoever, and that's obviously a big advantage there. We just can't make the isogenics, but otherwise certainly it's an important tool to address sporadic ALS. And it'll come out in some of the later recommendations.
But that's really Nicholas Maragax from Johns Hopkins. I agree with Jean-Pierre. That's really the power of using iPS cells, because they're these are non-familial. So you could argue to forget iPS cells from SOD1 patients, but rather just study the mouse. So it's really, we should emphasize sporadic ALS of a significant number to make one comfortable that there's a potential effect there.
We have comments from methodology.
So rationales are incomplete. First, an inference means that you have other premises that you're making an inference from, and you only have one premise. Plus, you're supporting a large number of recommendations with a very small rationale, and so it's hard, for example, to see why should you have dose response. There's not a direct link. You can't explicitly see why you're making that recommendation. So I think much of that needed stuff is in your background, and just as you organize that, it would be great.
Okay.
And then, of course, I will say my mandatory passive ways.
Okay.
Thank you, Brian.
Okay, so there are definitely some overlaps in the next presentation. Do I advance the slides, or? Yeah. Okay, so we were asked to really address the question of which models are appropriate. So the background to this obviously has already been mentioned, that the models that we have historically used have been exclusively based on genetic subtypes of ALS. And we have to acknowledge that the applicability to the vast majority of patients of these models has still to be really proven. So the comment earlier about iPS cells from sporadic patients is clearly very important, and they are certainly tools that should be used. So we've had a variety of models historically. We've had in vitro models that can be transformed cell lines with various genetic manipulations. They can be primary neuronal cultures from rodents, for example, or more recently induced pluripotent stem cell models, or ES cell models. The in vivo models can range through the whole animal kingdom up to non-human primates. So there's that background of complexity, and so the rationale really, number one, is that we agreed and discussed that each of these models is simply a tool which has its strengths and has its weaknesses. So we recommend that ALS researchers should use a particular model specifically for its strengths. So, for example, you might use high throughput modifier screening in certain lower organisms. But if you want to understand the engagement of your target, you'll want to do that in human cells and in a mouse model potentially. So our second rationale is that we think that models are a valuable tool for gaining insight into the biological events in the disease, so particularly early disease. So that's when therapies might be expected to be most helpful. So rather than focus on endpoints like survival, we recommend that ALS researchers should use models to understand the molecular events which are going on in the disease. And those molecular events can then be validated, ideally in neuropathological tissue or human tissue. Our third rationale is that we recognize that current models are exclusively based really on familial ALS mutations, and whether they're generalizable or not to sporadic ALS and other forms of familial ALS separate to that particular mutation is in question. And therefore, the ALS community should prioritize the development of models which, in as much as possible, recapitulate the features of sporadic ALS, for example, and particularly cytoplasmic TDP43 mislocalization, as a priority. So obviously we're not suggesting that there's an exclusivity here, we're suggesting this is a prioritization. As has already been mentioned, we recognize that induced pluripotent stem cell models have some drawbacks. So at the present time, the current problem is the variance between different differentiation protocols and reprogramming protocols is potentially greater than the variance between the disease and normal biology. And if you've got that situation, you have a problem. So we recommend that there should be some uniformity in how these cells are studied to allow cross-comparison. There should be reproducible differentiation protocols, a minimum number of 4 subjects per group, and isogenic correction as a control mechanism to try and remove the degree of variance between the experimental systems. And in particular, these cells should be rigorously karyotyped because we know that instability in reprogrammed cells can be very considerable. Rationale 5, in vitro cell-based models typically are based on mostly on one cell, sometimes on 2 cell types, neurons and glia, for example. And therefore, the complexity of the disease in the different cell types it involves is missing. So we recommend that ALS researchers should move towards developing in vitro models of multiple cell types with 3-dimensionality and complex organoid-like tissue models, and that will allow some of these difficult issues to be mitigated. In rationale 6, we believe that when you make a model based on a genetic mutation, you should try and recapitulate in as much as possible the architecture of that mutation, the genetic lesion, and that means, so at the moment there are, and the model should have some sort of disease-relevant phenotype. So that's a rather open-ended rationale, but it's there because the problem at the moment is there are no models that can completely do that. So we recommend that ALS researchers should focus on generating animal models with realistic genetic lesions. That means knock-ins or genome-based back transgenics in multiple different species, and to minimize the chances of creating biological epiphenomena which arise through overexpression. That is the last recommendation. Jeff's going to disagree with something that he was involved in writing.
No, that was super. That was really perfect. So I just want to bring up one small thing, and it harks back to, many of you read the Henrietta Lacks book about HeLa cells. So HeLa cells really started this huge biology of studying cancer, and they grew really fast, and everyone started using them and studying them, and by the 1960s, many other researchers found that other cell lines from cancer would grow really fast and wonderful, use them as tools. What they didn't realize is that they had contamination with HeLa cells. So a lot of that research was thrown out. What I'm getting to is that I completely agree with using iPS cells, but since they all look the same versus mice from different backgrounds, can look, you have a brown mouse, a black mouse, you know they're different, the cells all look the same under the microscope. So this may be digging a little too more into the weeds, but probably making sure you have the real genetic mutation if you're studying them, validating, not just karyotyping, but validating that's what you're looking at with the microscope is the right cell.
Small point.
Okay.
I don't feel comfortable with the wording realistic genetic lesions.
Yeah.
Because maybe we will never be able to make knock-in mice with the disease as a model, because rodents seem to be far less sensitive to what's going on. So shouldn't we say relevant or something like that?
So it's, I don't know, I mean, it's an aspiration. I mean, it's not a rule, it's a recommendation, because if you start off by compromising that, then you're already distorting the biology of the disease. I think it's the history of mouse models. And so if you start off with something that actually has, in as much as possible, the same genetic architecture as the human, well, that's a good place to start.
The thing is that we might disagree on this, and it's not because a mouse expresses something that it is a useless model. That's the point I want to make.
So I think we're not saying that overexpression models are completely out of the question. It's simply that, you know, you're going to get more chance of finding something disease relevant if you avoid that, is the sort of emphasis, really. I mean, none of this is meant to exclude any approach anybody wants to take. It's simply a way of trying to say, well, how do we get to where we are today, and how might we improve that in the future?
Just a brief comment. There is a very good model for ALS. That's the dog model with the E40K SOT1 mutation. It's the endogenous dog SOT1 mutation. It's perfect.
Okay,
no
so
one is using it. We have it in Sweden, and I believe it's also in Missouri. And the spinal cord of that is full of aggregates of misfolded SOT1.
okay.
No one is using it. I don't know why.
Thank you for that. Yes.
Hi. Sorry, so Rebecca Blunder from UCL. I just wondered if I could clarify a couple of things with two of the recommendations. So just going back to recommendation 2. Sorry. So I think in section B, it's suggested that we should be able to confirm molecular pathological changes in human tissue. I just wondered about whether we should kind of clarify the fact that when we use some of these model systems, as you said there, in fact, we're looking so early within the disease process that it may not be possible to confirm some of those molecular pathological phenotypes when you look at post-mortem tissue, which is maybe 70 years removed from those earliest events. And so, for example, demonstrating TDP43 inclusions, which would be possible to do in post-mortem tissue, but more difficult in, say, an IPS model. So I just wondered about whether we could make that a more sort of lenient in a way that you don't have to absolutely...
Well, it says ideally, I suppose, so it's you know, allows you not to have to do that. It's not a must.
Okay, fine.
And your second point?
Yeah, the second was, so recommendation 5. So yeah, I think that's brilliant that we absolutely need to improve the kind of IPS models that we have and using more 3D structures. I just wonder whether we still haven't validated about the cellular maturity of kind of organoid-like structures and the 3D structures. So just, I think, kind of initial stage will be to prove with direct comparisons of 2D versus using 3D scaffolding for IPS culture as well as organoid structures. So validating that there is definitely...
Yeah, and then again, these are emerging technologies, really. So this is a rather high-level kind of suggestion that they might be useful rather than a prescriptive way about how to use them, really.
Okay, thanks.
I just wanted to make a comment that actually the dog model is being used. We're testing the EAT2 PET tracer in that. It's being used for an antibody therapy and it's being used for antisense therapy.
So thank you so much for both Amelie and Eric for helping to put this session together. And there is a lot of overlap in themes between our outcomes and the ones that were just presented, so I'll try to skip over the redundant pieces. The one thing that is different, though, is you'll notice that our session doesn't have a question. I think everybody in the room would agree that rigor in all types of preclinical and clinical studies are absolutely required if we're ever going to develop an effective treatment for ALS or any other disease. So I think that the concept of the background here is the fact that we have seen lack of translation of potential therapeutics in ALS and other diseases as one used from preclinical models into the clinic. Some of that is done to poor study design, underpowered studies, lack of characterization of models. And I think that that's really where we need to start if we're going to effectively bring treatments towards the clinic. So the rationale here, which was kind of talked about in the first two, is that technologies are allowing models to be developed at a rate that was unimaginable even a decade ago. CRISPR technology, zinc fingers, talons, are facilitating the development of animal models from genetic studies. The sophistication by which we can express those transgenes is increasing with tissue-specific promoters, inducible promoters, cell-based systems such as IPS lines and 3D printing are starting to develop three-dimensional structures that one day may help us make neuromuscular junctions in a dish. So all of that needs to be characterized very carefully. And so the first recommendation really says that as a community, we should commit effort, time, and funds to characterize and validate the molecular, cellular, and if applicable, behavioral phenotypes of any new model that's being developed. And that ALS researchers should include controls when characterizing the phenotypes of these models. And we've talked a little bit about that in the first two panels, such as isogenic lines. They're actually getting fairly easy to make nowadays with things like CRISPR technology. So an example of this, if you wanted one that, may be relevant, is the fact that some of the early TDP-43 models, mirroring models that came out, were challenging to use. I mean, there wasn't a mention that the progressive NMJ loss in some of those models wasn't readily apparent like we see in some of the other models. So spending the time and effort, I think, to really characterize your model is critically important as we think about using them as tools for drug development, because, as Jeff said, they are just tools. It's not the disease in a mouse, it's a tool. Rationale 2 is more about the documentation of the process and to try to do a better job there. So adhering to some type of a best practice guideline for the community when you're thinking about bringing a new model into the community is really important, because as those models get distributed out to other researchers, you're the ones that know the nuances of them. You develop the model, you publish the first papers on the model, they get deposited in public repositories, and then other researchers license them and use them. But having some guidelines on the nuances we think is critically important to be able to replicate data. So the recommendations here are that all researchers should develop and publish best practice guidelines for the use of characterized models for preclinical research. ALS researchers develop best practices for colony management for rodent models, for instance. ALS researchers organize workshops on an as-needed basis to develop animal model best practice guidelines. And in the case of the SOD1 model, again, a tool, there's been multiple national and international best practices meetings. There's been multiple publications from lots of folks in the room on guidelines and best practices on how to optimize study designs for this model. So we should commit ourselves to utilizing these guidelines when we're designing preclinical studies. Rationale 3 is about, again, the concept of resource sharing, which I just mentioned, and that's to, as you develop new models, is to make them available so other researchers can utilize them as tools. So deposit model systems into public databases like the JAX, develop NTAs with your universities to get those models out into the world so that people can effectively use them for drug development, seeing whether we're going to move things forward quickly. So the recommendation here is that ALS research should deposit ALS animal models, ALS patient-derived cell lines, technologies around 3D printing for specific lines would also should be included in here, into public repositories following initial characterization. ALS researchers working with human cell lines should ensure that the appropriate language is included and informed consent so that we actually can share the models across the research and clinical community. Rationale 4 is that you need to utilize some of these tools for both studies with pharmacokinetics and pharmacodynamic measures. They're needed as we think about translating potential therapeutics in the clinic. we need to understand if the drug meets its target tissue. Does it bind to the target? Is there target engagement? Does that binding the target result in a change in the pathophysiology that you're interested in? These were all the concepts of what the recommendations were for this particular rationale. So the recommendations were that all researchers should develop models to look at biodistribution, determine pharmacokinetic properties, and candidate therapeutics under investigation. ALS researchers identify pharmacodynamic targets and confirm effects on these targets that are consistent with the intended pathophysiology. ALS research should examine dose-response relationships with a minimum of 3 doses in preclinical in vivo and efficacy studies. Number 5 was really about the statistical methodology in your designs. Often we do experiments in the lab without thinking about the underlying statistical analysis that one's going to do later. And I think the concept that Eric was really trying to hammer home here was that you should think about those things up front, much like we do in clinical trials. You should do them in preclinical studies as well. So ALS researchers should apply appropriate experimental design and statistical analysis to all preclinical efficacy studies and even other studies that we're doing in the lab, both with cell-based models as well as with in vivo models. And rationale 6 was independent validation of candidate therapeutics would enhance our confidence. Obviously, this is challenging. It's economically challenging as well as scientifically to replicate studies. But the more data that one has in this phase of drug development, the more likely your potential therapeutic is going to translate into the clinic. And even though it's economically a hurdle, let's face the fact, this is the cheapest part of drug development. When we're talking about working in the preclinical space with cell lines and animal models, even though the models might get expensive, it's the cheapest part of where you're going to go subsequent to these studies. So I think it's really important. So the recommendation here is the ALS research community should commit effort, time, and funds to independently validate preclinical studies of candidate therapeutics and publish both the positive and the negative results, ideally in peer-reviewed open access journals if we can. And I think that was the last one. Yeah. One more. Rationale 7. Again, more about reporting. So I think what we're thinking about here is the transparency in reporting the results of these studies. And so, again, it kind of overlaps some of the previous things, but ALS research should adhere to general and, if available, model system-specific reporting standards for key methodologies and analytical information about the preclinical studies done in research publications. I think that was the last one. And I'd open it up for questions.
May I ask you a question, Peter? This is an excellent overview of a lot of material. In recommendation 6, when you talked about preclinical validation, it brings up an issue that has come up several times already here with respect to whether results in one laboratory should be confirmed in another laboratory, or whether results in one model should be confirmed in another model. Could you speak a little bit about how much validation you are recommending?
I mean, ideally both, but again, I think time and money becomes the issue here. I mean, again, I'm not trying to say anything about ALS TDI, and Bob Braun, if he was here, would say the same thing I'm about to say. We try to replicate published studies as often as we can, but it is expensive. It's often challenging. It's sometimes hard to do the experiment exactly the same way, but if you can replicate a study in another lab with those nuances, that actually adds even more value and credibility to the fact that you might actually have a therapeutic that's worth moving forward. So, you know, I think that if you see a published study or a presentation where somebody has presented something that you have expertise in, and it's easy for you to try to replicate that work because you're interested in that target or you have a similar drug class, it would behoove the community for you to try to replicate the study and present it. And that way, promising things will bubble up. It's not going to occur by coincidence if 4 labs work on the same molecule and it bubbles to the top. It's kind of like the papers that came out around the nuclear pore stuff with Jeff's work and the Balo group a year or so ago. You know, it's not by coincidence that replication happens.
I'd say hallelujah to the issue of replication and rapid verification. My question's kind of related to that, and it's more, should we be trying to conduct mouse studies a bit more like a human clinical drug trial. So some of you might have noticed a paper published last year in Science Translational Medicine from a stroke group who took an antibody approach, used a MCA occlusion model, where I think it was 4 groups had actually shown a positive effect of this antibody. One group had shown a negative effect. So they took a new design and they actually ran it like a clinical trial, and they had 6 centers with small numbers of animals, and the data was all fed into an independent statistics group that were completely blinded to the data. And I think it's an interesting approach. I just wondered if the group had actually considered things like that.
No, we had not considered that. It's an interesting idea.
Oh, by the way, they found that the drug was negative.
Logistically, that's pretty challenging. Economically, it's probably also challenging. But would it improve data quality? It probably would.
Yeah, I'll ask a new question. I'll follow up on that. You know, sometimes when you talk to drug companies and they tell you about a new lead, and then you'll talk to another company, they'll say, well, we tried that drug. It didn't work in our hands. But that's all private tests. We don't know that. So replication is not just an issue among academics. Yeah. It's within an industry as well. That's actually not why I'm asking a question. So one, this was really great, Steve. But Bob brought up an interesting point that people say, they mumble about, shouldn't we try your a candidate drug in multiple different models? And the knee-jerk is to say, oh, that sounds good. But each model is built on a very specific molecular mechanism. So if I had a drug for C9, I said, oh, look, it works with a C9 mouse, whatever that might be eventually. It doesn't matter if it works with an SOD1 mouse, because it was very specific for C9. Yes. So we have to be careful about, and I know you didn't put that language
Kevin said that explicitly, though, Jeff, right? This was more about design, statistics, rigor, and best practices.
Yeah, and I agree. I'm just sort of commenting. I shouldn't, but...
Yeah. No, it's absolutely true. You shouldn't test your therapeutic hypothesis in a model that's not relevant. If your drug is targeting neuromuscular junction occupancy, don't test it in a model that doesn't lose neuromuscular junctions. If you're testing a drug that, targets neuroinflammation, you should look and make sure that neuroinflammation is present of your model. You should understand the kinetics of it too. When does the neuroinflammation start? When does it come up? What tissues? I mean, obviously a lot of logical answers to Jeff's thoughts there. Thank you.
All right. It's my pleasure to speak here among esteemed ALS researchers, but a little bit pressure for me as well, but I will do my best. Okay, so our group number 1, question 4, discussed what are a sufficient body of evidence to justify human trials. So we are talking a little bit more about the translational in the preclinical studies. So the backgrounds. ALS animal models mimic many, but not all aspects of the human disease. For example, SOD1 mice model do not develop TDP-43 proteinopathy. None of the TDP-43 mutant mice exhibit full spectrum of ALS features. Of course, prior to clinical testing, efficacy data obtained in mammalian model should be considered. However, rodents are species with much shorter lifespans than human beings. Furthermore, the effects of transgene overexpression per se must be considered, as it may contribute to the observed phenotypes in animal model. So the rationale 1. Many animal models that express ALS-associated gene mutations have been developed, but none of these models replicate the human condition fully. But many of them exhibit some of the pathological changes observed in familial and sporadic ALS. So, robust effects of a therapeutic approach on behavioral phenotypes, which is mainly the motor deficits, and on the attenuation of pathological changes in animal models are needed to justify human clinical trials. And this is the rest of the rationale one. A range of experimental readouts such as survival, functional, histological must be incorporated in preclinical studies, since no single measure can be expected to predict clinical efficacy. With positive effects such as a small extension of the survival or a small delay in disease onset in one model should be a matter of concern. Weak effects is interesting on condition that they are reproducible. So we set the recommendation 1. These are the conditions for human trials. Number 1, ALS researchers should incorporate a range of experimental, which is survival, functional, histological readouts in their preclinical studies, since no single readouts can be expected to predict human clinical efficacy. Number 2, ALS researchers should demonstrate efficacy in a spectrum of animal models to provide the high confidence for therapeutic activity in human ALS. ALS researchers should replicate positive animal model studies before initiating human clinical trials, so which is very important. But however, animal model efficacy testing is not necessarily a prerequisite for human clinical trials, because there are some occasions that animal model testing is not feasible. So we put that sentence, though it might be a little bit contradiction, but so the rationale 2 and recommendation 2, those are the generation of the animal models. There are published consensus guidelines by European ALS-MND groups, 2007 and 2010, for preclinical animals research in ALS. The generation of new animal models should be a priority in the field to overcome some of the limitations of the existing animal models and will represent specific ALS disease phenotypes or patient subgroups. So we set the recommendation 2. The ALS research community should prioritize the generation of new animal models. So rationale 3, this recommendation is about the selection of the therapeutic approach. So to date, the most effective therapeutic approaches such as shRNA, antisense oligos, and immunotherapy in the transgenic SOD1 mice are those that target mutant SOD1 itself, the cause of the disease. The development and model testing of therapeutic approaches that target causative factors such as level of misfolded or aggregated proteins, DNA, RNA repeats, correction of gene mutations should be encouraged. At the same time, ALS research community needs to keep in mind that the causes of ALS in the majority of patients are still unknown. So we set the recommendation 3. ALS clinical trialists should prioritize candidate therapeutics that target fundamental causative factors of ALS. And finally, that's the last slide for me, and we talk a little bit about the biomarkers in preclinical setting. Pharmacodynamic and target engaging biomarkers are very important in terms of that, which tells the differences between the animal models and patients. So we set the recommendation 4. ALS researchers should develop pharmacodynamic and target engagement biomarkers for candidate therapeutics to facilitate the assessment of treatment effects at the molecular level during clinical trials in humans. That's all from me. Thank you for your attention.
May I ask you a question about recommendation 1? The last part of recommendation 1 says that animal model efficacy testing is not necessarily a prerequisite for human clinical trials. Does that mean that we can test drugs in humans with no preclinical evidence whatsoever?
It's very difficult question, and this is, I think, the guidelines, and we want to make it the limitations for the possibility. So that's one of the reasons, and I think Jeff has something about this.
I think you've been hearing a lot in the others, and throughout all of these about the use of other tools that have emerged since 1995, and obviously a lot of people are focusing on iPS cells. The C9 antisense trial is fully based on iPS cells, not animal models. That doesn't mean animal models aren't good, and Steve beautifully outlined why the models are useful for pharmacokinetics, etc. But the primary decision to take a drug from to patients, does it require a mouse study? And I think the answer is no. It requires some preclinical data, but it doesn't have to come from mice. And again, historically, we've used other tools prior to the mouse models of ALS. Really was always never studied in a mouse model. Not initially, not to come to trial.
So should we say something about the importance of cellular or other preclinical data in this regard?
I think, as you pointed out, I think recommendation 1 is sort of contradicts itself. So maybe it can be reworded where you say ALS researchers should replicate positive animal studies before initiating human clinical trials, and then just add if it has an appropriate physiologic mechanism, and then you can leave out the last sentence about it may not be necessary.
Yeah, I will agree with that. Thank you.
I just think it's just an observation to add to the reason why I think a mix of the animal models, probably to what Steve Perrin's comment was. For example, the C9 model, where there's now a lot of debate whether a knockout actually could have some toxicity, and there's some interesting data of Bob's, if he was here, that came out today about the inflammatory pathway, etc. So it at least will inform, so it might be target dependent, that we might want to see, even if there isn't a phenotype or an equivalent model system that models a disease, it might tell us something about if you're going to do a knockout or lower it. There might be important things to think about how you're going to do that antisense technology. So just a comment there.
Great, thank you.
So one quick comment to Jeff and Bob's question is, you do need some animal model data to get into the clinic. You can't go from a screen in the dish. I'm not saying it has to be in the SOD model, but you have to do some of the things we've talked about for 2 days. 99% of chemical matter is so toxic, you can't put it into a person. So you have to do some pharmacokinetics, some tolerability, some biodistribution, some target engagement. You could do it in a wild-type mouse as long as the target's there, but often you need a diseased mouse because that changes the properties of your target engagement, your dosing, and your PD outcomes. So you just have to be smart about what tool you use, but you do have to do some in vivo work.
So Steve, I agree with you. All I'm trying to address, maybe you weren't addressing it to me, is that you don't have to prove clinical efficacy in a mouse, but you do need that biological data.
Totally agree. Thank you.
I really enjoyed your talk. In determining what preclinical models are necessary, should there not be some qualification in terms of the safety of the compound? For example, whether or not you have to go to a primate model if you're using something completely novel that's never been tried in humans before versus a drug that's already accepted and that we know has a safety profile? Should we not put some qualification? For example, stem cell infusions, we may have to go to a primate, antisense oligonucleotides for the first time, as opposed to something much more safe?
Yeah, I agree. So maybe we should put something about the safety, including the primates and the,
ALS is the kind of disease where the threshold is pretty low, given how bad it is to try things in humans, but some qualification would be helpful.
We should see the safety of that. Thank you.
I should add, and as I said in my introduction, we really focus on scientific premise and safety talk studies. I mean, no doubt they're essential, so we can build that in, but we just didn't focus on that.
Yes, about recommendation 3, maybe it's too strong to say that aggregation of TDP43 is causative. I mean, we have no enough scientific evidence. I mean, it's a marker of, but we are not sure that it's causative of motor neuron loss. So I don't think that it's a good thing to let it in the recommendations. I don't know.
Thank you.
Thank you. Great talk. And I just have one question. If you decide to do an animal testing, it's not a prerequisite for human clinical trials, apparently, according to the recommendation number 1. But if we do it on a mouse model, what is the bare minimum of readout changes that you consider encouraging to put it on the human trial? Because there's multiple measures, but is there a minimum really or no?
Well, that's a good question. So that's why we set the recommendation that the efficacy in the range of experimental readouts and also the spectrum of animal models. It's ideal, but I think we should think of these kind of things always. That's an important thing to do.
Dr. Jeffrowski will present the last clinical subgroup.
Just advance it. Oops, there we go. I got it.
All right, well, thank you. We're almost at the end. And the last group, it was really led by Michael Swash, Pam Shaw, Dr. Bano, and myself. Dr. Swash couldn't be here because he's ill. He has the flu. And so I'm stepping in for him, and that's a bad step in because I'm shorter, I don't have a really good accent, and I'm about 20 IQ points less than Michael. So I'll do my best representing Michael. What you're going to hear actually is, as you can tell now, we've been repeating ourselves in different ways through the last several research questions. So this last one is going to overlap probably at least 80% with all the others. So I'll try to move fast. And it's a little bit of an amalgam between about animals and then leading back to humans. So some of the latter questions will address, well, what can we do to human? What should we learn that we should be doing better in humans because we've derived things from mice, and you'll see that. So we've already heard multiple times that the vast majority of sporadic ALS trials, because the vast majority of trials we carry out are really for our sporadic ALS patients, have been based on the SOD1 mouse model, a fantastic clinical model of the disease, but there are many differences I think we've all come to appreciate as we've seen failures and therefore pick at this mouse, which is still a wonderful tool. And that, of course, it has many copies of the human gene, which our patients, not all of them do, but most of the ones we use, they're typically published on for drug studies, do have many extra copies of the mutant SOD1 protein. The pathology, although clinically these mice look partly like our patients, the pathology certainly does not. At least in some of the mice, big vacuoles, big, huge protein depositions that are not seen in most ALS patients. And for example, of course, you've heard this before, don't have TDP43, which all sporadic ALS patients have some abnormalities in. And of course, we all know that mutant SOD1 accounts for a very tiny percentage. Now, it's easy, I have to say this, it's easy in retrospect to do all of this, but when these mice were built by Tipu Sadiq and Mark Gurney and the group at Hopkins, this was a revolution in animal models. I'm defending it a little bit because it was the first real tool that we had to look at the human disease, sort of actually, other than cell cultures and cultured embryonic motor neurons that people like Chris Henderson used to use, so we thought it would be a great tool. And we spent years using it as a tool to study mechanism, and then later to study therapies, which obviously has been a bit of a disappointment. And so the facts, and I think Steve has pointed this out far better than we do here, the problems, of course, have been the poor rigor in preclinical studies, too few mice, balanced male versus females. We know in the SOD mouse that you can have differential effects with the same drug in males versus females. The lack of blind, tiny statistically significant effects have been often reported. There must be 100 papers out there that say the mouse works, and it's a 3-day increase in survival.
And again, this has evolved over time. The thought that a longer increase in survival somehow is better for patients, yeah, there's no data to support that anyway at all. And of course, as Steve pointed out, rare reproducibility. Now, new clinical studies are being focusing on specific genetic phenotypes such as SOD1, C9orf72, and there are other mouse models out there. Profilin's become ubiquitous too. They're for rare versions of ALS, but there are other tools beyond the SOD mouse. Very few of these, of course, have been used for drug studies because the bulk of the data that we have about these mice don't hold a candle to the huge studies, and especially those that have come out of TDI, where thousands of mice have been used, and you really have a sense of reproducibility of studies in preclinical. And there's no question the future studies are being planned using the mouse, but matching the mouse to the target. So antisense to C9, CRISPR to SOD1, shRNA to SOD1, and antisense to C9 and others, where you're really matching it. And I think that just that you've heard from all of us is that appears to be the real future. That's not facts, I know, so don't criticize me. That's an opinion. Okay, so the rationale number one. Most mouse studies administer treatment prior to the disease onset at early disease onset. But of course we know that human ALS is diagnosed at least a year or more after onset. Treatment trials often begin up to 2 years after onset. And the therapeutic effect, which typically is really more protection, not curing an animal, is less likely is felt to be less likely at a late stage of disease when more than 50% of motor neurons are lost or dysfunctional. And some drugs, frankly, are metabolized differently in mouse versus man. Vim showed that a number of years ago. So recommendations for rationale number one. Multiple models of ALS should be generated. Doesn't say they all have to be used for the same drug, just should be generated. We should avoid use of only the SOD1 transgenic models in sporadic ALS mouse trials. We should try to match the model, whether it's cell or animal or other tools, fish, to the disease mechanism. For example, C9orf, TDP43, not 2. Etc. We should try to define actual human brain injury pathways with post-mortem tissue. Now, this was slightly highlighted in others, but many believe that we should re-engage the use of human tissue to validate a target, to know it occurs. Yes, there's caveats about late stage versus early stage, but that's the event going on in our patients when we see them. And other candidate models should be used. Cell models with loss of nuclear TDP43, IPS cell models, including sporadic ALS, C9, FUS, TDP4, and others that come to bear as we learn the mutations and build those tools. And then, and again, this is brought up in others, mixed rodent or human cultures, neurons, astroglia, oligodendroglia, microglia, as tools. So rationale number one. Rationale number two, disease onset and severity. The rationale, human clinical trials include heterogeneous clinical phenotypes. Survival duration can be long or short, varying distributions of weakness, arm, leg, non-defined upper or lower motor neuron involvement, and overall lack of attention to therapeutic or pharmacodynamic signals. Current ALS clinical trial designs are sometimes relatively insensitive. Again, that was the rationale coming from human studies thinking about mouse. Recommendations. We should match with clinical trial measurements to parallel preclinical studies such as tissue and human cells. Better tools and observation for early disease at the earliest stages of the disease should be used, including quantitative EMG, cortical stimulation, cortical hyperexcitability, and imaging inflammation. Rationale number three, preclinical biomarkers. Again, this has been really heavily discussed in other sections. Rationale, biomarkers for diagnosis or progression are disappointing and are necessary. Neurofilament proteins are nonspecific to CNS damage. They're not diagnostic of ALS. They're not a sensitive measurement of rate of progression. CNS hyperexcitability is an early finding in ALS, but not specific to ALS. Recommendation. Tracking studies to evaluate drug actions or efficacy should be used during trials and evaluated in preclinical models. Again, we know we've discussed this already. Non-imaging approaches with PET or MRI for inflammation, microglia, astroglia, for early readout of efficacy. Need for motor neuron-specific injury imaging tools. Other CNS disease specific, such as CSF biomarkers, repeat dipeptides for C9orf72. So again, preclinical biomarkers. Again, we've obviously discussed this. And the last rationale deals with something we haven't talked about much, and I'm not sure, quite honestly, we've addressed it thoroughly here, disease initiation and spread. Little is known about what initiates ALS and how it spreads. Recommendation, we should consider carrying out trials on pre-symptomatic individual with known mutations, the idea of early on following patients, again, matching mouse to human or other models, including electrophysiologic measurements, markers of dipeptides, or SOD1, again, preclinically, and we know that some studies are being done. Emerging motor neuron injury, CSF markers, we should be looking for those, and PET MRI for microglial, astroglial changes. And that's it. Jean-Pierre.
I think that was excellent, really. Great job. The point about testing compound at different stage, I think, in the animal is very important. With minocycline, for instance, the results have been retested in the mouse model, in the SOD1 mice. When initiated early, at early stage, it confers some protection, but when initiated after what is called the onset of disease, 90 days, 100 days, it accelerates the disease. It causes more astrocytosis. So I think if we had known in those days, of course we would have recommended to be careful about the human clinical trial, but that wasn't known at that time.
Thank you.
Jeff, so another feature of the analysis of mice is that often the analysis is done as survival from birth rather than survival from disease onset. And in humans it's measured from disease onset. So what we're seeing often is actually a delay in age of onset with no change in survival if it was measured as a human disease. So I'd like to see a recommendation that the analysis also matches how it would be done in humans.
Very good. And others have actually promoted that. I believe Don Cleveland really makes a big point about that. Those who have found positive results in mice tend to look at that, and it is matching humans. You're right, Ben.
Just in wordsmithing, I think we should add neuronal and vascular to the last line.
Okay. Okay.
Also, I think we should make a difference between a study which tries to show that a certain pathway is involved in the disease by using an antagonist from a true preclinical study. Because in the first setting, you might treat the animals from before symptom onset, but it's not the same as saying that that drug is a candidate drug for therapeutic use. And then in the recommendation about, I think it was number 1 or 2, this one.
Yeah.
I know you might kill me, and others in the room might kill me.
Never.
Should kill me, must kill me, must consider to kill me. The iPSC model is a very new model which we don't have too much experience with yet, and we should not overdo it, because maybe in 5 or 10 years we say, well, it wasn't that useful after all. I'm out of here.
No one's going to kill you. Not now. I think, well, that's the whole point. I believe this is the point. We're making recommendations to think about in the future. This is clearly the new tool. If I had my own 2 cents, we've had SOD mouse, and it's a good tool for 25 years. Well, obviously, we're not sitting here with 15 other drugs, so I deeply believe we must consider other. This is we haven't codified this, though you see it in all the recommendations, that we really must embrace other models, and hoping that this will come back 10 years from now and have many other things we can change and not worry about that.
Yeah, well, it was great to hear those 25 years put out so eloquently. Just in the pre-symptomatic in your last one, you say should consider carrying out pre-symptomatic. I mean, they're sort of underway anyway, so it's just a bit of wordsmithing.
Yeah,
being done.
And it's also very clear that many of these overlap. I'm guessing whoever does this kind of stuff will blend and merge all of these, and we won't have constant repetition. All right.
We're going to move on now to a very simple charge that was given to Eric Macklin to review the appendix on biostatistics in the consensus guidelines from 1999 and to advise us about how this should be altered going forward.
Yeah, thanks, Bob. So Group S, which is not for sleep time, although it's getting late in the program. So there's one individual item that we were asked to look at, item 8, trials should be designed with careful and detailed statistical analysis, which should begin in the planning phase, see Appendix E. So we're proposing to expand that a little bit. Investigators should design trials to align trial aims, study collection and plan analyses. Investigators should consider statistical issues to minimize bias and maximize efficiency. Investigators should prepare a detailed statistical analysis plan prior to enrollment for open label trials or prior to breaking the blind for blinded trials. And then again, we have this somewhat potentially voluminous appendix that we would propose including. So the original Appendix E opens with a couple paragraphs and sets its scope just to address pivotal trials, identifies the importance of collaboration among trialists, statisticians, clinicians, suggests that design should be tailored to the question asked. We'd propose expanding that to include additional work outside of the final pivotal trial, presumably including preclinical as well as all phases of clinical research, and propose including patients as important contributors to considerations of proper design. Then there's a sequence, and these are E1 if you're looking through page 70, 87, and 88 in your handout. Goes through a number of bulleted items or paragraphs. Dropouts is the first, suggesting we should try to minimize them. I don't want to read through these individually because I like to take advantage of everyone's time here to suggest where we could make improvements rather than just what was there before. But so just to identify, we would suggest trying to expand that to address appropriate methods for handling informative censoring, due not only to death, but as I wrote this up, there are also, I know, a number of situations in which participants will withdraw consent for further participation prior to their death, but with death relatively imminent in their course. And those need to get handled very much the same as an observed mortality event. Stratification, we talked about that a good bit on the first day, and would make sure in this section to identify that stratification in this context is related to randomization structure, and would propose expanding discussions of alternative randomization schemes. I think my primary suggestion for improvement in our handling of this would be to focus our attention for stratification on those measures that are predictive of the outcome under study in whatever design was being pursued. Ideally, that would exploit as much information as we can glean from baseline characteristics of those participants, perhaps using mechanisms like Origen's predictive modeling. It could be at the other extreme as simple as just if you're a de novo diagnosis, then you're likely to have a more rapid course than someone who's already had the disease and has survived for 3 years. So the things that we have a lot of intuitive understanding of, we can either formalize those into a predictive model and stratify on our predicted outcome. That would be the most formal, or at a minimum, identify that the stratification is to homogenize the expected outcome in patients. There's a section on adjustments. With our expanded scope to earlier phase studies, I'd suggest that we need to provide some lower bound to avoid overfitting. We don't want a study of 20 individuals where we adjust for 10 different parameters, obviously. So a little bit of nuance there. And in particular, although the sample size might accommodate it for later phase studies, I think in the interest of emphasizing generalizability of our results from later phase studies, we'd probably be interested in less adjustment. For analysis, the original guidelines were written primarily in the context of pivotal trials with survival endpoints. With our expansion, we'll be emphasizing more efficacy outcomes, which would presumably, emphasize in our efficacy outcomes measures that are rates of progression of functional outcomes or strength or other more fine-grained measures, with a focus on mixed models and other mechanisms, statistical models for handling covariance among repeated measures. My pet interest is in having us acknowledge that site-by-treatment interactions are a component of our multi-center trials that we tend to ignore, and by assuming that they're zero, we overestimate our precision in our estimates, and that's another source, I think, of where our phase 2 trials often fail in phase 3, is we think that we can expand to 15 sites from our first 4 without acknowledging that our uncertainty among the treatment effect of those 4 was not properly accounted for. Time to event, again, was a focus originally, and we expand that to consider a wide range of outcomes and discuss the appropriate selection of outcomes for analysis and the incorporation of patient-reported outcomes. Lead-ins was an idea that was raised and is still, I think, in need of study to identify whether it is justified. It's obviously a large devotion of resources, substantial additional costs to the study and effort on the participant side, particularly if the lead-in is used not only to stratify participants, but in order to establish inclusion criteria. If you run a lead-in and then half of your participants you decide are not eligible, that's you've engaged the participants in a lot of effort without providing them very much gain for their efforts. Surrogates, as a part of the original recommendation, and as we've talked about before, we really don't have lacking an effective treatment in the disease. We don't have the framework that will allow us to make a validation of any biomarker as an effective surrogate. So I think we'll probably be revising that merely to say that surrogates are not currently available, and these are the criteria that we required to validate them, in reference to the FDA, and I apologize, probably EMEA and maybe Japanese regulatory guidance as well. So there's a number of additions, and these are the pieces I want to spend slightly more time on, and then elicit your brainstorming with us on what more might be usefully included in these. Data standardization is an important component that wasn't touched on originally. So the recommendations, bulleted there: use of standardized and validated data instruments in cases where they're available, use of NINDS common data elements, use of standardized coding. I guess it doesn't need to be MEDRA and WHO drug coding, but some standardized dictionaries of adverse events and medications. Use of CDIS compliant data collection. Alex is giving me the quizzical hand wave, but we would certainly benefit from standardization, because the next point that we wanted to make is that we should also be emphasizing the importance of data sharing, and data sharing is very difficult if we don't have standardized data collection. CDIS maybe is not the end-all be-all, but it is one framework that could be a foundation for effective data sharing across studies. A lot of data has been contributed to PROACT. The piece that I might amend to that is the second bullet, that we need to ensure that these shared data sets, although anonymized, retain enough information to make appropriate use of them. PROACT, for instance, doesn't retain year of data collection, and so it's very hard to make an evaluation of whether use of those data as historical controls is still useful to us if it's a mixture of 1995 data as well as 2015 data. Country of origin and site, again, I think could be just not to know which site, because that would be anonymizing, but to be able to recognize aggregates of participants who came from a site, I think could be an important point of analysis as we try to use those more broadly. Trial registration needs to go somewhere. I'm not sure it has to go in the stats section, but sort of stats related would be an advocacy for open submission of protocols and statistical analysis plans, in addition to pre-registration of the trial itself with a regulatory body. More stats related, group sequential designs for all but earliest phase studies. I think there's a good reason to take advantage of well-developed statistical theory that allows us to make some interim analyses of data in one of two ways. One, to facilitate not continuing to expend resources unwisely when there's already evidence of futility. So an interim futility analysis for even moderately sized studies, I think, has some good basis. And consideration of sample size re-estimation, using fully blinded data and pooling all into a single sample causes, imparts no penalty with respect to type 1 error, so there's really no cost to it. It provides you some information. It lets you know whether you're on target for identifying the type of treatment effect that you set out to identify, or whether you're off target, and updating. So nuisance parameters is a bit of a catchphrase. That would be the variance in a continuous measure, would be event rates and time to event data. Pre-specification obviously is always essential in these, and there's existing extensive guidance on these techniques. Historical controls is a more difficult topic to work through, but in the interest of trying to be most efficient in our work, we should not blind ourselves to the the large volume of existing data that we already hold on to. When we embark on a new study, we should be learning in a more formal way from what we already know. Obviously, every time that you sit down and think about a new study, you're drawing on a vast amount of previous experience with patients' course, with disease response, with drug interactions. But there's a lot of other formal data that statisticians usually don't get their hands on to incorporate into the analyses, and we can be more efficient. Whether that's, I think, rarely going to be pure use of historical control data, but there may be instances where we can increase our efficiency by augmenting a concurrent control set of concurrent control data. Bernard mentioned yesterday some negative experience with relying too heavily on historical control in a small concurrent sample. So we don't want to undermine an effort by relying too heavily on it. But I just can't imagine that we can't find ways to more intelligently use the data we have in hand. Pilot and exploratory studies, Charity has a paper. There's actually a relatively dearth of publications out there trying to provide good guidance on how to appropriately design pilot and exploratory studies. Often people will go into them with just a rule of thumb of, oh, I studied 10 people or I studied 30 people, and so I must have, by that volume of data, have enough to answer my question. So we'd like to, again, try to, in the interest of being more hard-nosed advocates, advocate for clear specification of what is it that you can reasonably learn from a pilot study, and is the data that you're going to collect, will it answer those questions, and either help the study team revise its goals so that they're more aligned with what's feasible to achieve, or ideally come up with a design and a planned sample of data that will allow you to reach a go, no-go decision when appropriate, that you have high probability of going forward with a drug when it's successful, and yet you have relatively high probability of stopping a drug early when it's not successful. And being very clear about what those no-go criteria are and the probability of reaching them under a set of reasonable scenarios is, I think, an important point that's often not pursued enough in pilot studies. Subgroup analyses, there's a lot, again, this is in the same spirit of drawing on information that we already have in hand. Once you've completed this study, well, some pre-specified subgroup analyses, which would be warranted, and then hypothesis generation from subsequent analysis after the fact. That's what I got. Thank you.
While people are going to the microphone, I will just say that as a clinician, non-statistician, this is superb. You've added things that I think have come up in the meeting are very important, and you've changed things that I think are important from before. So
thanks, Bob.
Well done to you and your friend.
Thank you, Bob. I guess I cannot pretend to be a younger investigator, so I have to write it down. So first of all, CDISC, I just wanted to comment that CDISC requires registration that costs a lot of money. So we can pretend that we're CDISC compliant in the sense that we can use some standards proposed by the CDISC, but to register the disease, it costs hundreds of thousands of dollars with CDISC consortium.
Well, we welcome your guidance on thinking through those issues.
Right. So absence of dates in PROACT data set is required. It was required by legal requirements of the donors, data donors. So we can revamp this, or we change the legal documents or basis for this. But for now, we cannot do this.
Yeah, I think I would hope that there's some granularity of data that we could reach agreement on. That
everything is available. It's up to us, but I believe it should be renegotiated with the donors.
Yeah.
The third thing about PROACT, I would love to see a recommendation not only about publicly sponsored trials, but also about private trials, to at least to recommend them to donate their data, the data sets into PROACT database. And another one, I was wondering, I believe I didn't miss it, about utilization of predictive models as possibility for either decreasing or eliminating placebo arms.
Was that a question?
Predictive model like we used
yes,
for
yes.
yeah.
Yeah, no, I'm definitely an advocate of that.
So Eric, I just wanted to ask you about, I got a sense of a judgment on lead-in trials and using that to help with predictive models, whether that's a positive or negative. Basically you said, well, that's not necessarily good for the patients because it may exclude patients after they've put a significant amount of effort into it. But in fact, it may help patients in the long run by choosing the right population to do your study in.
Certainly.
And that way you actually get a result at the end that is a little bit less ambiguous than it might be. So I think it's something that we should not discount. I think it's something that's we should consider strongly.
Right, I probably was stumbling on my feet up here, but because I do believe that there's value to it, I think maybe we just need to be clear with our participants about what the whole enterprise is and how we're going to make use of their commitment to us.
I think we mentioned earlier in other sessions the point of having the GUI link to the data. That's still going to be in our guidelines. The GUID.
GUID? I'm sorry, I missed that.
The GUID is the patient identifier, unique patient identifier. Wasn't that one of our earlier in the session today, we talked about recommending that be one of our guidelines?
Yes, so adding that into the data collection component, yes, right. Good recommendation.
Now the other question I had with respect to the top of your slide and the bottom of the slide, everything at the top you're taking out? Or?
No.
Okay, good, because I wanted to make sure we kept pre-specified analysis plan and all that kind of stuff.
Right. So the revisions were meant to be identifying where we might change what was there, and not that we would be eliminating what was there.
I thought it'd be nice to see something on clinical staging. There's various clinical staging methods. Some of them are being validated, and it'd be quite a good way of either measuring severity or using as an inclusion criterion or potentially as an endpoint for it.
Yeah, I might be interested to chat with you more and understand where they can best be put to use. Okay.
Excuse me, could you predict a little bit more or expand on the predicted outcome? The predicted outcome, could you expand a little bit, because this is a very crucial issue.
Right, so I'm drawing on the idea that Origin's trying to commercialize that using baseline or lead-in data, say 3-month history, that they can come up with a prediction of your outcome with perhaps 20% accuracy. You know, the metric during the PROACT competition was that the best algorithms could explain about 20% of the variation in ALS FRSR scores 9 months out. And I believe Origin's doing somewhat better than that, but that's somewhat our framework, that with some with some baseline data, an appropriate algorithm can make a prediction of an outcome measure somewhere in the future with some degree of accuracy, obviously not perfect, but some degree of accuracy. And that's then an appropriate basis for stratifying participants, so that those who are expected to have the worst outcome in 9 months could be lumped together as one stratum, and those with expected best outcome in 9 months would be a different stratum, and we could ensure that there's not chance confounding between predicted disease severity and treatment allocation in a randomized trial.
What elements do you have in your hands to validate these models?
I'm sorry, say again. What evidence?
What evidence you have behind these in order to see if your predictive models are validated?
So maybe I'm more, blasé about it. But if these prediction models are completely inaccurate, then all we've done is randomly assigned participants. To the degree that they provide any accuracy in prediction, we've ensured some avoidance of chance confounding. So I don't think there's a lot of downside to their use, and they do provide some potential upside. Is that responsive to the question? No. We'll chat afterwards.
I think, Eric, I think I can provide what our plan is for that. Dave Ennis, Origin Data Sciences. I think sort of the first step in validation is to set aside part of a PROACT and then validate the outcomes that were predicted for that set-aside group using the algorithms. The second stage is to take an external data set, preferably one that's a recently obtained data set. And so we've done those first two steps. And then the third step is really kind of the toughest hurdle for us, and that would be to make predictions today truly today for patients that we have no idea what the outcomes are. And that's the effort that we are currently organizing. So we've gone methodically through a stepwise validation process of the algorithms. And therefore, functional rating scale, FVC, and survival, and really it all depends on the data set that we're presented with, the types of outcome predictions that we can make. So I think we're starting to really come to the ultimate validation of these models.
Did you publish this or not?
We are preparing it. Kind of the initial contest was published in Nature Biotechnology of January of last year, but we've improved the models substantially since then. So we are preparing the publications.
I could just add that those of us, quite a number of groups now, have begun to test the waters with Dave and his team at Origin. It looks very promising, is all I can say at this point, but the data that we've seen so far and the experience of working with them suggests that there may be real promise here, but as you can hear, more needs to be done. And I think it's great that we are open to integrating that as it appears more promising.
So, Bob, you have a Word document of this current, all the publication. So if you don't, so if you have, it would be very convenient for Eric to revise it.
Oh, I already have it as a document. Yeah.
Entire document.
Well, Appendix E.
Right, that's excellent. Okay. Yeah. So before Bob is going to give us instructions what to do for breakout sessions, I'm sure you are dying to get out to get sunshine. So we propose to, in 10 minutes, to get outside. You see the big stairs, and everyone stand there, and we'd like to all the pictures, all of us. So then I'll go back to coffee and breakout sessions. You have all names, which breakout room on the table. You have the instruction. There's the 2 rooms different, right? Right. So don't get up. Bob is going to give the instruction what to do with the breakout session. Then I'd like you to come to stairs for taking pictures. Thank you.
So Hiroshi, back here. We're half an hour early. What time should the breakout session start? 3? How about 3?
Half an hour before.
Yeah, half an hour. So 3 o'clock?
So, well, if we have time. We have also coffee break, but we have to hear Bob's instructions. And also, Group S should go to Delphi methods, right, Gary? Group S, statistics, has to go to... Right, okay.
So if I could just make a couple of remarks about...
Hold on, please sit down.
If I could just make a few comments about the breakout sessions. There have been many comments made following each of the presentations, but in addition to that, there have been emails and there have been comments made by the people on the webinar. So there has actually already been some feedback, and group leaders are encouraged to take that feedback to their groups. The breakout sessions should highlight those bits of feedback that might impact a revision. The idea of the time in the breakout sessions is to use the feedback from Gary Bronseth, our methodologist, and the other feedback that we just alluded to for making revisions to primarily your recommendations, but to some extent also the rationale. So the main focus is looking at those recommendations and revising them together as a group to include all of the sources of feedback that we've talked about. By the end of the breakout session, the hope is that you've done all the tweaking that you think you need to do so that your rationale and your recommendation are pretty much in final form. They'll be presented tomorrow morning, and all we want to hear tomorrow morning are the recommendations and any comment about where there still may be controversy or why certain changes were made. Any question about all of that? Right. So there are 4 of us who will be spreading ourselves out among the 8 breakout sessions. Hiroshi, Ben, myself, and Gary will each be assigned 2 of the groups to interact with, and we will be trying to be helpful.
So what is the strategy to avoid all the repetitions?
What do you mean repetition?
Because, for instance, the biomarker problem came back in almost every session. The redundancy is probably the proper word.
So for now, I mean, it's an important issue long term. For now, if you have repetition within your group, take care of it. But if it's repetition between groups, don't worry about it. We'll catch that at the very latest with the line station.